So hi everyone. Today we are very excited to have Dr. Olivier Elemento from Cornell. And just to give you a quick biography of his impressive background, Dr. Elemento received his degrees from France, and his most advanced degree is doctorate in computational biology from the University of Montpellier, CNRS. After that, he completed his postdoctoral training at the Louis Sigler Institute for Integrative Genomics at Princeton. And then after that, he joined Vail Cornell Medicine in 2008 as a faculty. And since then, since then and more recently, he's been the director of the Carly and Israel Englander Institute for Precision Medicine. So it's actually a very exciting institute to bring multidisciplinary research for precision medicine technologies and informatics. It's exactly actually the spatial omics as well. It has both technology and informatics to uncover the molecular mechanisms of disease and individualize disease treatment and prevention. He is also Associate Director of the Institute for Computational Medicine, Director of the Laboratory of Cancer Systems Biology, co-leader of the Genetics, Epigenetics, and Systems Biology program in the Mayer Cancer Center at Vail Cornell Medicine. And he's a tenured professor in the Department of Physiology and Biophysics. And without further ado, I'd like to turn to Dr. Elemento.
Thank you. Again, thanks so much for the invitation. I'm very pleased to be here today. I want to tell you about the research we're doing in spatial omics using the imaging mass cytometry technology that we've acquired now a couple of years ago. And I think, as you'll see, hopefully, really is giving us very exciting data. I'm hoping that we'll make this interactive. I don't know how you do this usually, but please do feel free to interrupt. It's so much more fun, if we have a discussion. I'm hoping we'll have time for questions after the talk itself. But do feel free to interrupt. I don't know that I will see the chat, but if somebody is monitoring the chat, please do interrupt and ask any questions. Happy to pause and again have a discussion. So let me just start with a couple of disclosures. I've started a couple of companies that essentially kind of focus in the precision medicine sort of field. Some of them are using this kind of technologies that we're going to talk about today. Companies have been more focused on AI and in kind of sort of some of the aspects of what we do, but keep in mind that there's some potential conflict there. And also I have some additional conflict that you see in this slide. So the talk today is going to be focused on imaging mass cytometry, and I'm pretty convinced that many, if not all of you, know what the technology is about. I'll just recap very quickly, just so that we're all on the same page. This is a technology where you take tissue sections, you label these, basically stain these tissue sections with antibodies that recognize different protein markers or basically markers that antibodies recognize. What's special about these antibodies is that they are conjugated to metals that have a very specific mass, and therefore whose mass is kind of detectable using a mass cytometry device. The principle of the imaging mass cytometry device is that you take the tissue sections that you've stained and use a laser to ablate the surface of the tissue sections. And the product of the ablation, the ionized metals, go into the site of basically the mass cytometer for quantification of the abundance of the different metals. And the quantification of the abundance of the metals basically gives you a quantification of the abundance of the different proteins or different markers that the antibody recognize. And that gives you essentially maps of where the markers are present in the tissue sections. So the idea here, what obviously is very special about this technology is that you co-stain with up to 37, 38 markers at the same time. And therefore, if you have markers that are recognizing different cell types and different functional aspects, signaling aspects, of each of the cell types, you can really understand, the sort of tissue architecture and function of specific cell types in these maps in a way that you couldn't before. I should say that this technology is single cell resolution and therefore really gives us a lot of insight about tissue architecture, proximity between cell types, and so on. I'm going to try to give you a sense of some of the analysis that we're doing to kind of exploit this information, and obviously understanding that we're just scratching the surface. There's a lot of really interesting computational analysis that can and will be done on this kind of data. My sense is that the computational methodologies, when you compare, for example, with fields like single-cell RNA-seq, probably a bit sort of behind. There isn't the kind of standardized software and packages that you see in single-cell RNA-seq, for IMC, but that's coming, and I think that that will democratize even further the technology, and I think will make it even more exciting. So this is what the technology looks like here. This is the Hyperion module that is connected to the Helios, which is a traditional CyTOF sort of module. This is the kind of tissue sections that we analyze. Basically, sort of tissue sections we have to sort of identify or isolate or kind of basically look at specific regions of interest. It's not a technology where you can scan the entire slide, unlike other technologies that maybe can do sort of not as powerful in terms of number of markers. So you have to tell the machine where to look. To identify regions of interest, as we say. And so that's something that is important as part of the process. We work with pathologists, at Cornell, to identify those regions, because you have to make some choices about what you want to look at. Typically, we do this, as you'll see, across many samples, and we try to pick a good number of representative regions so that we can sample the heterogeneity that you see in the same tissue at different locations, and that's something that obviously is important to keep in mind. So I'm going to talk about two different projects. The first project is a project focused on B-cell lymphomas, which is something we've been working on for some time in a variety of different ways with colleagues here at Cornell, including Dr. Melnick. We've been working on the lymphoma projects. We've been doing lymphoma sequencing. We've been doing mouse modeling. We've been doing all kinds of analysis of lymphomas. There are different types of lymphomas. There are aggressive lymphomas, which is the ones that we're focusing on today. There are less aggressive lymphomas called follicular lymphomas, which we have samples we want to analyze, and we haven't started this. There are some interesting, I think, differences in terms of tissue architecture between those two. So the follicular lymphomas are a bit more like normal lymph nodes sort of tissue. And I think that, as you'll see, I think some really interesting sort of architecture. These aggressive lymphomas are not really supposed in theory to have a lot of, sort of architectural features, which I think makes them really interesting to analyze, because actually we do see interesting architectural features. It's just they were not really anticipated based on how people looked at the tissue, in the past. We're scratching the surface there, but I think you'll see, I think there's some really interesting things that come out. So we've been looking at two different panels of antibodies, as you'll see, looking to analyze 600 aggressive lymphomas. We've already have data on 200 of these lymphomas. These are DLBCL, so they're called diffuse large B-cell lymphomas. And we have typically 2 punches for each of a tumor. We do this in TMA, tissue microarray format, which allows us to process a lot of tumors at the same time. And therefore kind of improve the throughput. One thing about these tumors, they're coming from a collaboration with a leukemia molecular profiling project, lymphoma leukemia molecular profiling project, which is a consortium that is kind of standardizing a lot of analysis, including genomic analysis. So we have, in addition to tissue, we have genomic information on these tumors as well, which is obviously a great opportunity to kind of correlate some of the architectural features, the cell type abundances that you'll see with mutational content of tumors themselves. And as you'll see, we're seeing some really interesting correlations already. So without further ado, so these are the panels that we used. As I said, we're using 2 panels here. And this is what the content of the panels and the metals that each of the antibodies are connected with. As you can see, the different panels have overlapping markers. Lots of markers of B cells, of B cell function, but as you can see, also lots of markers of T cells and NK cells, and including some for example, phosphorylation markers, histone modifications, which really works really well, as you'll see. transcription factors, and so on. So it's a pretty broad array of different markers that capture different aspects of the biology of these tumors, as I said, that we know reasonably well. Some of these markers also have therapeutic targets. We're very interested in identifying novel therapeutic options for some of those patients. Some of these tumors are very aggressive and hard to treat using chemotherapy. So we're always looking for additional potential therapeutic targets. So we actually did put therapeutic targets on the panels like CD73 or CD47. This is kind of, just to get a sense whether, what are the cell types that express those markers? Are there indeed differences between patients? Are there subgroups of patients that express those markers that would potentially make them a good candidate for a clinical trial, which has not been done for those yet. There are some checkpoints, as you see here, and so on. So it's a broad range, as you see here. This is the kind of, this is just to show you the tissue microarray format. Basically, multiple calls, different tumors, and kind of laid out in the kind of an array format, if you want. And as I said, we typically have 2 calls per patient. This is a format where essentially you have all the calls are kind of embedded in Paraffin, and you can essentially take slides, cut off slides from the microarray and do if you want multiple consecutive rounds of analysis on adjacent consecutive slides. As you know, the analysis of the Hyperion analysis of these markers give you this kind of representation here. This is one spot on the tissue microarray. This is looking at 3 markers out of 37. As you know, the challenge of this technology is that you can't visually, I think it's hard to visualize more than 3 markers at the same time, because otherwise, the colors, sort of start, overlapping, sort of too much to visualize anything. But what you see here is that you have PDPN, which is a lymphatic vessel marker, which, clearly you can see the structure of the lymphatic vessels in this analysis. You have CD8, which is a T cell marker, and you can clearly see the T cells that express CD8, and CD68, which are macrophage markers, and you can see clearly that those cells are quite visible in this kind of analysis. We do a lot of QC, and part of it is making sure that we look at tissue and tissues that we know well. For example, normal lymph node tissue do have the right or the expected cell types according to the detection of the markers that this analysis provides us with. And that's the case. The normal tissues actually have quite a bit of very sort of almost stereotyped architecture. For example, there's germinal centers in the lymph node and surrounded by cytotoxic T cells. The germinal center that you see here has multiple regions: the dark zone, the light zone. So that's, we use that in some ways as kind of QC, where we expect to see certain cell types, and obviously the markers that represent those cell types have to show up for us to declare that the antibody is doing what it's supposed to be doing. And the detection of them is also kind of as accurate as we like it to be. So that's an important aspect of the whole process. Then we get into the analysis. So the analysis, as you know, it can be a little complex, but I've been worked on this technology for some time. We've developed some good methods for doing this. The first thing to do, which we have DNA markers, is to detect the cells, and we use, as I said, the DNA markers to do this. That allows us to identify lots of different cells. The project that I'm going to talk about today, this 200 DLBCLs, gave rise to about 3 million cells. So it's a lot of cells that we have to analyze using this technology. But once we found the cells, then we go back to the different channels of the different metals and essentially quantify the abundance of the different markers within each of the cell. So if you have 37 markers, each cell now is represented by a vector of 37 values, which corresponds to the intensity of the markers at those cells. We end up with sort of lots of, as I said, about 2 million cells, more than 2 million cells that have sort of 37 values attached to them. We cluster those cells with the idea of looking for groups of cells that express the same markers, which we basically sort of think of as cell types. They define groups of cells that express specifically the same markers. And that allows us to, as I said, find the cell types. In this analysis, this is variable, this is slightly older, but we find in the order of about 28 different cell types. So 28 groups of cells that expressed the same combinations of markers in some expected cell types and some less expected. As I said, because we have information about the genetics of these tumors, so we have the mutational profile of the tumors, one thing that we can do is a little bit, like I said, also, maybe QC. There are certain mutations, like mutations in EZH2, which we've contributed to kind of describing in the past, that are actually pretty abundant in these B-cell lymphomas. It's about maybe 20% of B-cell lymphomas have mutations in EZH2. These are gain-of-function mutations. So EZH2 is in charge of depositing, catalyzing H3K27 trimethylation. We know that tumors that have EZH2 mutations have more K27 trimethylation. So then we can look at the tumors that we have and ask whether there are tumor cell populations according to the cell type identification that we have that express H3K27 trimethylation in those tumors. And as you can see here, it's very clear that the tumors that have mutations in the tumor cells in EZH2 do have a lot more K27 trimethylation expressing cells as a whole. So that's a good validation also, again, about the ability of the technology to find these populations and then to sort of define them in a way that we expect. We also see, as we expected, that tumors that have mutations in EZH2 actually do have additional B-cell populations that have lower expression of MHC molecules. So we published a paper actually a couple of years ago, actually a year ago, showing that indeed EZH2 is kind of shutting down expression of MHC molecules and therefore enabling immune evasion. And that's exactly what we're basically seeing here. Increased abundance of populations of tumor cells that have low MHC expression in the mutated tumors. So that's all good. I think this is very reassuring in some ways, that the patterns that we expect to find, to show up. But we're also finding a lot of patterns that we didn't quite expect. And I think the ones that are interesting are the ones that kind of correlate mutations in the tumor cells with behaviors outside the tumor cells in the microenvironment of the tumors. For example, we see that tumors that have CD79B mutations, if you look at the tumor cells themselves, I'm not showing it sort of here, but they actually don't necessarily have, well, they have some specific phenotypes. But I think the most striking phenotypes are actually outside the tumor cells. The tumors where you see those mutations have a high abundance of these CD3 positive, CD8 positive sort of T cells, and especially the ones that have exhaustion markers. So basically it shows you that there is, most likely some kind of crosstalk between tumor cells that have the mutations and the microenvironment that results from being surrounded by tumor cells with those mutations. And I think this is exactly what we're able to find here. We've been trying to generalize this analysis to kind of look at all possible crosstalks between mutations, which is on the x-axis here, and then populations of cells according to the Hyperion analysis. And as you can see, there's a lot of correlations. The red is positive correlations, meaning if you have a mutation, you have higher abundance of a particular sort of cell population, and the blue is negative correlation, meaning if you have a mutation, if a tumor has a mutation, you see less of a particular cell population. And as you can see, really, this map is showing abundant crosstalk between the tumor cells and that have specific mutations and the microenvironment. And we're exploring now, specific sort of patterns, but I think this is giving us a lot of material to work with in terms of understanding this crosstalk. Lymphomas can be categorized in specific subgroups. This is purely a genetic sort of type of categorization. A combination of mutations that has been shown to create subgroups or subtypes of lymphomas. And so that's what we've been able to do here, is to correlate or categorize each of the tumors that we have into the genetic subgroup. But the question that we ask, maybe related to the questions that I asked earlier, that I was showing you earlier, is really, is there a correlation, or is there kind of more detailed patterns of immune infiltration in specific subgroups? For example, what we see clearly here is that this subtype here, the MCD subtype of lymphomas, if you look at the abundance of CD8 T cells according to the Hyperion analysis, it's the subtype that has, on average, the highest abundance of CD8 T cells. Very clear here if you look at the other populations or the other categories. But as you can see, there's a tremendous range, right? I mean, it's not like if, all the tumors have high amounts of CD8 T cells. So we ask, is there something special about the tumors in this particular category that have high versus low abundance of CD8 T cells? And these are examples, pretty striking examples here of these two extreme situations where you have two tumors, exactly the same genetic subtype MCD. This one has a high amount of CD8 T cells. This one has very few. When we look overall at all the tumors that we have, at these two ends of the spectrum of CD8 T cells, because we have survival information, we can actually correlate these subcategories with survival. We see very clearly that the tumors that have in the MCD category and have a lot of CD8 T cells actually doing pretty well from a survival standpoint compared to tumors in exactly the same genetic category that have very few T cells. So what this is showing you is that these genetic categories that with people have sort of defined may actually have a lot more complexity if you were to look at the microenvironment in addition to just looking at the mutations. And that's something, again, I think, sometimes I show this slide and people say, well, look, you probably could have found this using IHC of CD8. And that's absolutely the case. But the reality is that nobody would, have, sort of thought to look at for, this kind of correlation. And so it's only because we did this in an unbiased analysis without a specific hypothesis initially in mind that we were able to find this correlation. And that's, I think, an important feature, for this kind of project. Going back to the to kind of more spatial analysis, now that we have a cell type for each of the cells, we can go back to the tissue sections and label each of the cell with the cell type that it belongs to. So that allows us to, sort of do this. This is an example here, one cell type at a time. But in theory, you can, well, in practice, you can really associate each of the cell to a particular cell type. And this is what we did here. This gives rise to this really beautiful map here where you have each cell as one dot, if you want. And it has a color that corresponds to the cell type that it belongs to because it's expressing markers that put it in that cell type category. And as you can see, this is an example of, this is actually reactive lymph nodes. There's actually a tremendous amount of structure in this kind of tissue here. You can see regions of tissue that have specific combinations of cells. Like here, the pink cells and the blue cells. Here, this is a region that has a lot of these, sort of greenish, cells. Here, you have regions that have maybe a combination of these yellow cells and maybe sort of these red cells. So there's a lot of structure here, and I think we've been trying to think about how we can find maybe sort of maybe better or sort of accurately define this kind of structure here. What we're trying to do is to essentially find communities, which is, we call them communities in maybe probably different ways of labeling them. But the idea here is to look for areas in those maps that have the same combination of, different cell types. And basically what we called, sort of communities, or I guess, you could call them cellular neighborhoods. And so we have an algorithm for doing this. We found, just a long story short, we found in the order of about 32 different cell communities across the entire data set. This is what these communities look like here on the left side. Again, this is a community is a kind of a combination of the same cell types within close proximity to each other. That's another way to define them. And one thing that we did is to kind of go back to the actual tumors and look at, how many communities typically a tumor has. And I think what we see clearly here, and maybe a bit unlike what I showed you for the normal tissue, is that, each tumor seems to have, mostly one community. There are actually some tumors here that have maybe a few communities, but I think the pattern that I'm seeing is the fact that every tumor has actually only one community. And so that's, I think, an important, sort of observation about, maybe the slightly sort of less let's say, structure maybe, that you see in tumors compared to what you see in normal tissue with multiple communities that coexist, with each other. And again, this is not, a very sort of robust sort of description, because there are tumors that have multiple communities, as you see here. One thing that we see is that there's also a correlation between, the community content of tumors with another way to define subtypes in B-cell lymphomas, which is more of a transcriptional sort of type of definition, an ABC versus GCB subtype. And clearly, there are certain type of communities that clearly correlate very strongly with a particular subtype, and we're exploring this, in more detail. And this is here the community content in terms of the major cell types, as you can see here. As you can see, most of the communities have a lot of tumor cells. It's interesting is that most of the communities that we find using this analysis, have a combination of tumor cells and some of the other cell types, but in different abundances, as you can see here. We've been looking at community content and correlation with survival. One observation, for example, is that tumors that have communities that have very few macrophages actually tend to do pretty poorly in terms of survival compared to other communities, which is something that we're exploring maybe a bit contradictory compared to what we've seen in other tumor types. This is looking at MHC expression, and this is looking in highly apoptotic communities. So the blue ones here are the communities, are basically the tumors that have a lot of these highly apoptotic communities. And as you can see, those are actually doing pretty poorly compared to the less apoptotic sort of tumors. So that's, again, patterns that we're exploring further, but I think are really quite exciting and interesting. I'm just going to switch gear and just show you another project which is quite different. This is a project focused on actually COVID. This is something that we started kind of as a result of having a hospital that at some point cared for a lot of COVID patients. Unfortunately, sadly, a lot of patients died, and before they died, they gave us consent for example, to get access to some tissue, autopsy tissue after they passed away. So we were able to get lung tissue from COVID patients, and I think a really an amazing opportunity to understand the the effect of the infection on the lung, which is critical site for COVID infection, understanding what it does to people. So we were able to get access to multiple sort of subgroups or patient populations, mostly in the COVID space, patients who died early of COVID and patients who died late of COVID. They died early not necessarily because of the lung involvement. Typically they died early for a variety of different reasons. The way we kind of segregated those two different groups here is because we think that it gives you a sense of progression of a disease. The patients who died late had more progression, and so a lot of damage, as you'll see, accumulated over time. And so we think of these two distinctions as a way to understand progression and advanced stage versus maybe earlier stage. And so part of this project was also to analyze control lung tissue from patients who did not die of COVID, who died of something else. Car accidents, would be one reasons, but also patients who had other types of respiratory infections, for example, influenza or pneumonia. So in this analysis, we were able to get in total about 237 images across all these number of patients. And should say, first of all, so this is a project with Rob Schwartz here at Cornell, Adam Borgzak, who's our lung pathologist, very talented lung pathologist, Hiran, who runs our Hyperion machinery, and Andre, who's a superstar analyst, postdoc analyst, who really has led a lot of the analysis, and hopefully you'll see, really is really pushing the envelope when it comes to analysis of this kind of data. So this is what we found. So we obviously this data is very rich. Actually, let me just maybe backtrack here and show some of the markers that we looked at. So these are the markers that we looked at here. Again, so these are immune markers, but also markers that are important when it comes to lung and lung biology, as like for example keratin, IL-6. These are some of the cytokine sort of markers. I can say different cell types that correspond to different different immune cells and structural cells and microenvironment, and so on. So these are the markers that we looked at in this particular analysis. We had markers that focused on the stromal compartment of the lung, basically that allowed us to quantify, for example, the amount of fibrosis that we would see in patients with COVID. We also had markers or let's say ways to calculate the kind of emptiness that you see in some areas of the lung, which in normal lung is actually quite strong. There's a lot of areas in the lung that do not have any cells. by looking at areas that don't have any cells and quantifying this across different groups of patients, including the patients who died late. You can see that very clearly there's like a decrease in the amount of these free regions, free of cells that go with severity of a disease. And the patients with advanced disease have essentially very, constricted sort of, lungs with very few areas that do not have any cells at all. We're also able to identify a fibrosis score by quantifying the amount of the sort of the signal that markers that quantify fibrosis. We're able to also quantify fibrosis. And fibrosis, as you can see here, is basically following the completely inverse sort of pattern with COVID patients who, especially those who died late, having a tremendous amount of fibrosis and fibroblasts in their lungs. So going back to this, this is kind of the same kind of clustering analysis that I showed earlier, which is a clustering based on cells that express specific markers that allows us to label each individual cell, give them a name essentially. And as you can see, these are the names that we are kind of trying to analyze. Epithelial cells, obviously, very critical in the lung. We also wanted to get a sense of dying cells that express specific apoptosis markers. These are immune cells, different types of immune cells. Macrophages, obviously, very important. Fibroblasts, endothelial cells, and so on. So these are the cell types that we analyze using this analysis here. So I mean, long story short, I think the punchline of one of the punchlines of this paper that's in review now or in revision is really that we see quite a bit of changes in COVID patients compared to normal tissue, as you would expect. This is not a surprise, but I think by kind of analyzing multiple cell types at the same time as Hyperion allows us to do, I think you get a picture that maybe is a bit more subtle than what has been seen before. One pattern that has been described pretty early is neutrophil involvement in COVID. And I think that's just something that we see here. When we count how many neutrophils we have in the lung of COVID patients, we see clearly that patients with COVID have more neutrophils than healthy individuals, the green ones here. But as you can see here, one is that that seems to be kind of a phenomenon that you only see in early COVID patients that kind of goes away in more advanced disease. And two, this is a phenomenon that's actually not particularly specific to COVID. And I think that's something that maybe escaped the attention of others who did this analysis without looking at the broader context. Neutrophil involvement, is not very COVID specific. On the other hand, if you look at things like macrophages, we see that very clearly, especially in advanced disease, the macrophage involvement is gigantic. This is actually probably what really characterizes the most COVID, especially advanced stage disease in COVID, is the extreme abundance of macrophage that you see in those patients. Mesenchymal cells, as you can see here as well, extremely abundant. And fibroblasts, as I mentioned earlier, is also quite a very strong feature of the disease. So again, I think this unbiased analysis of these multiple cell types at the same time, I think, is revealing patterns that again, were not necessarily always expected. And I think that's a very very important sort of utilization of the Hyperion. One of the things that you can do using this technology is to query also, proteins that may actually not be part of a host. So we actually were able to find an antibody that recognizes the S1 protein of COVID, and does that pretty accurately. As a result, what we can do is to quantify the amount of infection within each of the cell types that we found in the lung of these individuals. One of the things that is also helpful as a sort of quality control is the quantification of the amount of AS1 protein in all of this disease or healthy individuals. And as you can see, AS1 is just not there whatsoever. So that's reassuring. It's very abundant in early and late COVID patients, and that's basically what you expect. But now we can overlay the abundance of ES1 protein to different cell types. And there, I think there are some surprises that emerge. One is, and that's not a surprise, that's actually exactly what you expected, which is epithelial cells, do have the most S1 abundance. That's basically what you expect. This is a primary target of the virus. It expresses a lot of the ACE2 entry protein. So that's basically expected. one thing that we can do is to, because we have markers that kind of, we think are downstream, signaling markers, for example, we can compare the infected cells, which you see here, versus the non-infected epithelial cells. And as you can see, there's actually a lot of downstream activation of, both cytokines, but also, sort of other cytokines and, signaling markers, that you see only in the cells that are infected and not in the cells that are not infected. These are the same cell types, one infected, one not. And that's sometimes in the same patient. And so, again, a lot of differences that you observe, sort of here. So that, allows us to understand better the function of the impact of the infection. But going back to this map here, what you see is that actually quite a few macrophages do express the S1 protein according to this analysis, which is maybe a little bit unexpected, because we didn't really know that macrophages could be infected. Obviously, one explanation for this is that the macrophage just ate up an epithelial cells that was infected, and therefore, kind of positive, for that reason. But we actually looked at that specifically, and we see clearly that the macrophage that are positive for S1, they actually don't express the marker specifically of epithelial cells. So we do think that this is additional evidence. Obviously, there's probably potential additional explanations, but it's good evidence that the S1 the virus can infect macrophages. And we actually see that the virus in theory could potentially be infecting quite a few other cell types, including endothelial cells. They do express ACE2, but also neutrophils and maybe some monocytes and some fibroblasts. So that's a very, very interesting observation. And this is here kind of maybe demonstration of this, maybe sort of further analysis in that direction. This is looking at epithelial cells that express S1, and then as you can see, they're here. That's and sometimes they're kind of surrounded by macrophages, as you can see here. This is macrophages that seem to express S1. Clearly, also a good demonstration that it's the case. And here, this is fibroblasts that also express S1. So again, we see a little bit of everything by looking at these maps in more detail. One of the things that was also very strong, as I said, is the abundance of the apoptotic markers in cell types that you didn't really quite expect to have those. So what we see, for example, is that the apoptotic markers, such as complement or cleaved caspase, actually pretty highly expressed in some macrophages, as you see here. One surprising thing is that there's a lot of epithelial cells, as you would expect, the infected that express these apoptosis markers, but there's actually a lot of what we call kind of bystander effect, off-target effect, if you want. Basic apoptosis in fibroblasts, which are not infected typically, but clearly have a lot of expression of the apoptotic markers, meaning essentially they're dying. So this is, would think of, say, kind of maybe the consequence of tissue damage, bystander effect of, killing of adjacent cells, but not directly due to the infection, but essentially as a kind of indirect consequence of infection. And so I think the picture that we are seeing is basically, a lot of damage as a result of infection in cell types that you don't, not even infected themselves. A lot of macrophage involvement that are probably being recruited into the tumor to be able to maybe fix, the damage that's happening and maybe kind of make things worse as a result of, extreme recruitment, as that we see very clearly. One of the things that we've kind of looked at, and maybe superficially, but I think it's kind of interesting, is the this is a more spatial analysis where we look at proximity between cell types. We developed an algorithm that allows us to quantify the proximity between two cell types as a whole. It's not at a level of individual cells, a level of many cells together. Can we kind of say that some cell types kind of get closer to each other in a particular group of patients or get farther away from each other? We actually, we actually see a lot of kind of movement, if you want, of cell types, comparing, for example, COVID patients with healthy patients or healthy individuals. As an example, there's a lot of kind of increased proximity between neutrophils and macrophage in COVID patients. Just examples. We see also differences between patients early stage versus late stage. This is interaction between fibroblasts and macrophages, which you see here as one of the strongest sort of changes in terms of proximity as a whole And we see basically there's a lot of invasion of macrophages within fibroblastic sort of walls, alveolar walls, that illustrate this overall sort of pattern. And so I think some of the sort of remaining analysis, I think, more focused on trying to understand progression of a disease. When we put all of these region of interest, these areas that we scan into this kind of map, this principal component analysis, we see that there's very clearly structure. Each of the region here is defined by the abundance of the different subtypes that we found. So there's very clear structure, and the first principal component, in our view, is really kind of interesting because it kind of almost defines progression of a disease, where you go from healthy individuals to maybe what you see in purple here, which is early COVID towards late COVID. It's like a progression in this particular direction. Another thing that we did is a principal component analysis where we can map the importance of the different factors in terms of defining the subgroups or defining the groups. You see that there are macrophages, as I say, that are very, very important in defining the more advanced stage of a disease. But something that I didn't really emphasize, but in fact, instead of finding one group of macrophage, in fact, we actually do find different groups of macrophage, probably 4 or 5 different groups of macrophages. What's clear is that some of these subgroups of macrophages are clearly associated with advanced disease, but there are actually subgroups of macrophages that express slightly different markers that are actually not associated with COVID, for example, associated with pneumonia. So I think there's more subtleties in terms of the type of macrophages that connected to COVID that I think we need to delve into more details and understand better. But I think, again, this is a technology that allows us to do this, and really shows a very rich biology that I think is going to continue to reveal very important aspect of the disease. And this is showing additional sort of component. Again, once we define this principal component 1, kind of that defines the progression of a disease, then we can look at all kinds of different additional, sort of features. As I said, lacuna, which is empty regions of the lung that decreases with sort of progression of a disease. As fibrosis that increases. There's a complement activation that increases. This is a clear progression. In all these different phenotypes that you can observe using this analysis. So I'm going to stop here. Hopefully, I think we'll have a bit of time for question. But again, thank you so much for your invitation. I'm very, very happy that I got a chance to speak with you guys, and I hope that we can have a little discussion and continue this discussion. Hopefully, in a more interactive way. So thank you, guys.
Thank you, Dr. Elemento, for this great talk. We have one question on the chat. Would you like to read it, or should I read it for you?
If you don't mind, I just let me try to.
Sure.
I'm not sure to access.
You can go ahead and raise this, the full screen. I think that's.
Oh yeah, no, I got it now. Okay. Yeah. So maybe I'm happy to read it. So the question is, can you please describe the method used to determine cellular communities? So we're happy to do that. So again, this is an experimental method, not something that is necessarily very ready for prime time, but I can explain in just a few words. The idea here is to essentially have a window on each of the cell that we look at, and essentially what we do is to quantify the abundance of the other cell types within that window. So essentially, this cell at the middle, if you look at the neighbors, the composition of a neighborhood is a lot of these cells, and not so much the other cells. So each of the cell is described essentially not so much by the markers that are expressed, but by the composition of a neighborhood. And so what we do is define a profile of composition of a neighborhood for each of the cell, and then we essentially cluster the cells based on this profile. And then we, in addition to this kind of clustering, we basically attach proximity between, the sort of neighboring cell types. So we basically end up looking for clusters of cells that have the same abundance of other cell types in our neighborhood, but are also in close proximity, and that basically gives us the communities. So it's not, not necessarily, sort of perfect, but that, does the job at least, as a sort of first attempt. Second question from the same person is, what is the upper limit of cell type markers for Hyperion? So that's in the order of about 37, 38 markers. It's basically just number of markers that we can use. So that all being said, there are many, many more antibodies that you can use. If you have a good antibody that you can tag using a metal, you can use it for Hyperion, obviously after some optimization of different parameters. So the number of markers is very large, including, as I said, includes phosphoproteins, includes histone includes a lot of different things that you can raise antibody against. So there's no fundamental limit, except that at the same time you can only use 37 or 38 markers, and that number is kind of fluctuating as FluidDyme is coming up with additional sort of metals, combinations of metals. All right, so I'm going to move on to the next question. From Collapsed. Q1 is for spatial analysis of your tumor data, are you using custom scripts or software like Cytomap to identify regions in your tissues? The answer is, yeah, basically we're using custom scripts right now. We actually have been looking at commercial software, and I guess, we haven't really found anything that is very compelling so far, but we're still looking. But by and large, what we're doing now is using custom scripts. Second question is, how are you determining the number of communities of regions in your tissues? So I'm not sure. So the number of communities is defined by essentially how many clusters of, cells or neighborhoods we find using this analysis. Again, that's an approximate number because we can go at different levels of granularity, of course, as you can imagine. So there's a little bit of kind of ad hoc, sort of choices that have to be made in this kind of analysis. Number of regions of interest, ROIs per sample or per tumor, that's an important question, if that's what you mean. We basically looked at, as I said, in the B-cell lymphoma project, in the order of 2 regions per tumor, and we do see that in some cases, the content of those 2 regions are very similar to each other. In some other cases, they're not so similar, so that underscores the fact that there's tremendous amount of heterogeneity within the same sample. And I think that's something that we need to keep in mind. And I don't know yet exactly what is the optimal, number of regions to look at. I think it may be kind of, tumor type specific or tumor type dependent. But that's something that's very, very important to keep in mind. Another question is, can you also comment on the average cost of each assay per sample? So I'd say, part of, the cost that, we incur is really kind of operational cost. I mean, there are costs related to, conjugating the antibodies and, really sort of optimizing, the antibodies and the titration and so on. These are kind of fixed costs in some ways. But then, every time that we run the machine, we have additional costs, in terms of, argon, in terms of operators and so on. So I don't necessarily have a good breakdown of, the cost. I'd say, it is, once you have a panel that works well, I think, it is, reasonably tractable. I think it's, it's not extremely high. I think it's in the order of a few $100 per slide. So it is, tractable. Obviously, if you do what we do, which is we look at hundreds of samples, the numbers do add up. But at the end of the day, it is tractable. It really depends on the amount of region that you want to scan. I think that's the main driver of the cost. If you want to scan a lot of the same sample, it takes more time and therefore more money to do so. But if you choose your regions well and represent different aspects of different locations of a tumor, you can sample the tumor or the tissue that you're looking at in a way that can save you a lot of cost down the line.
So I have 2 questions. I think one is related to what you just described. We got to choose the region that can best describe the pathology here.
Yeah, exactly.
So I was very impressed. So I think in your study you were able to kind of link the genomics data or just a clinical genetics data and everything else. I think your center is collecting to the spatial data. But something like a large diffuse large B-cell lymphoma, the tumor is humongous, right? Yeah. So have you looked at different regions and so which can better kind of associate with the clinical cytogenetics data and so on and so forth. So you do see any kind of difference if you sample the center or in the peripheral region of that tumor?
Yeah, that's a wonderful question, frankly, and I don't know that I have a great answer for this now. As I said, per tumor, we typically sampled, 2 spots, 2 locations. They were chosen by pathologists, who say, well, look, this region is interesting. There's a lot of tumor cells. This one has, maybe, more of the immune cells. So, I think that, I don't know. I think to me, that's a very critical question that we need to address is, how much heterogeneity do we expect to see? What is the best approach to be able to capture that heterogeneity? Keeping in mind that, I think down the line, I think we'll integrate probably data across multiple different, sort of layers. One thing that I'm really excited about, because we're also doing quite a bit of work on analysis of H&E slides, kind of simpler staining, sort of type of analysis, is to combine these pieces of data onto each other, right? There's no reason to ignore the H&E stain, which is, as you know, gives you a lot of really good information about, the heterogeneity, about, about the, even, sort of abundance of particular cell types. I feel like we need to combine those 2 things, into one kind of global measurement. And the idea would be to sample, maybe sample specific locations in the slide so that you can maybe use the H&E of the entire, slide to reconstruct, maybe the signal across the slide. I feel like, there are some really nice opportunities to kind of do that. I think that sometimes, people are a bit focused on what they do and they kind of, maybe ignore some of the data that's just available. And H&E is available and scanned, in many cases. I think the future, in my view, is really to combine those 2 pieces of data because H&E is always there pretty much. If you have tissue, if you work in a pathology department with pathologists, they've done staining of with H&E and they, so that's available. Very powerful, in my view, to combine the 2.
Mm-hmm. I have another question, I think related to a COVID study, which is super fascinating. I think a beautiful work. So I think other people observe like a germinal center formation in a tissue, and somehow that's quite different from kind of normal pneumonia. Not normal, but just a non-COVID pneumonia. So have you kind of paid attention to that particular structure?
Yeah, unfortunately, not so much. I think we had a few, sort of B-cell markers here. We've been focusing in the lung, and I don't know. This may be a matter of where we looked, and we didn't really see a lot of B-cell action, if you want, where we looked. But it's not to say that if we were to look in other places, and we wouldn't be able to find. But that's I think that's a very important sort of comment about Yeah, it really, is maybe a limitation of, not limitation of the technology, but the fact is that, we have to choose, where you look in specific tissues, and we didn't really, start this project with that in mind. But I think now that, we know of, this reasonably recent finding, I think we should definitely explore that more and, and find out whether there's some kind of signal in our, in our data that maybe, explains, some of what's been seen before. That's a very good point. Yeah.
Mm-hmm. Thanks.
Quick question from here. So these are nice discoveries from COVID patients, but what do you think about translation of these results to diagnostics or therapeutics? Is there any pathway?
I think that's a wonderful, point as well. I think that, obviously, the lung biopsies is probably off the table, for most patients. That's something that is, easy to achieve. I think that what we're thinking about is to maybe translate some of these findings to, circulating markers or circulating cells, right? Things like, maybe myeloid cells, maybe macrophages, or, sort of, if you can see evidence of those in the circulation, they are much easier, much more readily quantifiable in the circulation compared to obviously the lung. So, I think some of the patterns that we see in the lung, we hope to be able to look for them in the circulation. To be honest, we were talking about the second Hyperion that we bought, kind of a justification for this, with some of the people who pay the bills were basically exactly that we could use, the site of, not necessarily only the Hyperion, but maybe the Helios as a way to, using the same kind of technology, more or less, kind of look for markers that we saw in the lung, maybe look for them in the blood. And I think that, there's a project that we're studying now. We'll see how that goes. But, I think that the patterns were very strong in terms of, as I said, sort of here, kind of a correlation with cell progression. We actually were able to build predictive models that, sort of predict, who's going to progress and so on. So, I feel like, we might be able to find early signals in the circulation that tell us that there's progression happening or, ongoing, in the lung. And that, would be one way to apply these findings to a more practical setting.
Exactly. I think that will be very exciting.
Yeah, I agree. Yeah.
Liquid biopsy and solid.
Yeah,
tumors.
absolutely. Yeah.
Any other questions? Feel free to unmute if you are about to ask. And then there's a note saying that Cytomap is a free open software. It might be helpful.
Yeah, that sounds great. Thank you so much. I really appreciate the link. Yeah.
Okay, I think since it's almost past 3, let's thank Dr. Elemento again. Thank you so much. It's clapping time.
Thank you.
And next week, we're going to continue with another human tumor atlas project using another technology called cyclic immunofluorescence. And the speaker will be from Peter Sorger's group at Harvard Medical School. And he's the scientist, Jiaren Lin, working in the lab for a long time and original developers of Cyclic technology. And with this, I think we are concluding today's talk and meeting. And thank you guys all so much, and hope to see you next week. And have a nice weekend.
Thank you, guys.
Thank you.
Bye. Take care. Bye-bye.
Bye-bye.